Near-infrared spectroscopy tissue oximetry is an emerging non-invasive method to evaluate tissue oxygenation. It can measure oxygen delivery to the cells of the brain and kidneys, two important vital organs. Though this technology was first described more than 40 years ago, use of NIRS has increased significantly worldwide in the past decade. NIRS is based on the fact that near-infrared light is able to penetrate biologic tissue and can obtain real-time non-invasive information on tissue oxygenation and metabolism. Oxygenation-dependent light-absorbing characteristics of hemoglobin in blood is utilized in NIRS. Cerebral oximetry, or measurement of oxygenation of the brain, has been studied most widely in the field of cardiac surgery. This is because these operations can lead to significant neurologic complications and associated morbidity and mortality. NIRS can also monitor oxygenation in muscles. NIRS monitoring in muscles has found application in the field of sports science. In cerebral oximetry, fiber optic sensors are positioned on each side of the forehead and covered by an opaque plastic patch to prevent ambient light. In adults with thicker skull, near-infrared light penetration into the cerebral cortex is only 3 to 5 millimeters, while in neonates with a thinner skull, it can penetrate 10 to 15 millimeters. In one device for cerebral oximetry, 4 different wavelength laser lights are emitted for maximization of accuracy. Reflected lights are captured by fiber optic sensors, and interference from tissues outside the brain is abstracted. NIRS is being used in thoracic surgeries, aortic arch surgeries, carotid endarterectomy, as well as abdominal and neurosurgical procedures. NIRS values have been correlated with reference values of cerebral tissue oxygen saturation derived from simultaneous radial artery and jugular bulb venous blood samples. Oxygenation of the brain and kidneys can be monitored during interventions in the cardiac catheterization laboratory. In a study, it was found that when a heart rhythm abnormality or cardiac arrhythmia develops, NIRS values fall simultaneously. When desaturation problem develops, NIRS falls 10 to 15 seconds earlier than pulse oximetry. On improving saturation, NIRS returns to earlier values 10 to 15 seconds before pulse oximetry readings. Thus, it may provide an early warning. A survey of the Congenital Cardiac Anesthesia Society in 2021 among its members showed the usage of NIRS by 34.7% in the cardiac catheterization laboratory and 97.1% in cardiac surgery under cardiopulmonary bypass. How is NIRS different from pulse oximetry? Pulse oximetry calculates the percentage of oxygenated hemoglobin in arterial blood. NIRS calculates changes in oxyhemoglobin and deoxyhemoglobin in the tissue under investigation, which contains both arterial and venous blood. First set of journal references. Second set of journal references. Thank you for watching this video. Please subscribe, like, share, and post your valuable comment below this video. Kindly press the bell icon after that for getting all updates.